Hi guys, and welcome to this cellular biology course. Today, we are going to discuss the main points about cellular biology. By the end of this course, you are going to know the methods for studying the structure of the cell. So let's start with definitions. Cytology allows the microscopic structure of the cell to be studied outside of any tissue organization. Molecular biology and biochemistry, functioning of the cell. Cell biology uses cytology, molecular biology, and biochemistry to study the cell. And we have a tissue, an assembly of cells filling a function within an organ. Examples: epithelial tissue, connective tissue, muscular tissue, nervous tissue. An organ, assembly of several tissues that fulfill a specific function in an organism. Examples: brain, liver, and pancreas. Studying the cell. Under normal conditions, the human eye has a separating power or resolution, a visibility limit close to 0.2 millimeters, 200 micrometers. At 25 centimeters, it can distinguish 2 points 0.2 millimeters apart. To increase the resolving power of the eye and study the cells and their constituents, microscopes are used. So we have two types of microscope: light microscopes or photonic microscopes, and electron microscopes or electronic microscopes. Here is an electronic microscope or electron microscope. Observation with an optical or photonic microscope. Working principle: uses photons as an energy source. The name photon comes from Greek and means light. Provides a magnified image through optical lens combinations. The eyepiece on the eye side 2 to 10 times, the objective on the object side 2.5 to 100 times. The variation of the magnification of the objective by that of the eyepiece allows you to obtain the magnification of the microscope. For example, 10 times eyepiece and 40 times objective equals 400 magnification. Components of the optical microscope. Parts of a microscope worksheet. Number 1. Eyepiece tube or body tube. 2. Nose piece. 3. Objective lenses. 4. Stage clips. 5. Stage. 6. Diaphragm. 7. Illuminator. Number 8. Eyepiece or ocular lens. Number 9. Arm. Number 10. Coarse focus. Number 11. Fine focus. And 12. Base. Specimens. We have two types of specimens: tissue specimens and cytological specimens. For a tissue specimen to be done, we usually use a biopsy, a tissue fragment, living being, or excision biopsy, sampling of the entire lesion, or puncture biopsy, sampling of a tissue, a core. We usually use needle or trocar. A cytological specimens by scraping, brushing, needle puncture, collection of washing fluid, collection of secretion, flow products, affixing a sample to a microscope slide. For tissue samples, preparation steps: levy, fixation, decalcification, hard tissues. We use nitric acid and EDTA, ethylene diamine tetraacetic acid. Dehydration, clarification, impregnation, inclusion, cups, deparaffinization, and rehydration, coloring, dehydration, assembly of the sections between blade and cover slip, and observation. So let's start with fixation. Preserves the structure of the dead cells. Fixers kill the cells by keeping its constituents in place, create very stable chemical bonds between cells molecules. Example, formaldehyde and formalin. Then we have decalcification, hard fabrics, tissues impregnated with calcium salts, bones, teeth have a rigidity which makes them unsuitable for microtome cutting. Decalcification equals process used to remove calcium salts from these tissues. Use nitric acid, EGTA, ethylene, diamine, tetraacetic acid. Then we have dehydration. Prepare the tissues for inclusion because paraffin is not miscible with water. Clarification. It's a passage in inorganic solvents. Can be toluene or xylene. Impregnation. Impregnation by pure liquid paraffin in a oven between 56 and 60 Celsius. Passage in 3 baths. Inclusion. Stiffens the tissue to make it easier to cut and prevent cell deformation during cutting. Paraffin 56-60%, L-bars, plastic molds, metal molds. The last 2 points are the tools that we use to do an inclusion. We use the paraffin and we use sometimes L-bars, plastic bars, or metal molds. They are all in the shape of L-bars. Deparaffinization and rehydration. Using a paraffin solvent like toluene or xylene, essential step prior to coloring because most dyes are hydrophilic. Decreasing alcohol baths, gradual. Then we have coloring. Coloring, it's a mean available to distinguish the various structures of the tissue. It highlights the elements in the chemical composition of tissue structures. Each tissue component has a particular behavior in the face of dyes. Increases the contrast between cellular structures. I think we're gonna stop here for the first part of this video and we're gonna finish in the second video, so stay tuned, stay connected to have more informative content as this one. I hope you've enjoyed the video and it was informative for you, so stay tuned for the next video and goodbye.